Hello, my name is Jorge Cole, Sales Executive at Hyperberry, representing the US and Canada markets. I'd like to welcome you all to another HPP Innovation Week session titled How to Validate Your HPP Product. In this session, we'll walk you through the procedures to perform a validation test, as well as introduce you to the Cornell's HPP plans and service that they offer. Guest panelists in this session include Dr. Vinicius Sarment. Vinicio is part of the hyperbaric food application and processing team, where he provides technical advice to clients and potential buyers of HPP industrial equipment in the American continent. In addition, he is actively involved in research and technology dissemination efforts related to HPP food processing. We also have Dr. Randy Warabo. Randy is a professor in the Department of Food Science at Cornell University. His innovative research has resulted in over 150 publications and led to industry adoption of several technologies like UV, HPP, and antimicrobial additive as alternative approaches to enhancing the microbial safety and the quality of food. I would like to hand the floor to Vinicio for this presentation.
Well, thank you, Jorge, for the introduction, and welcome everyone to our session. It's a pleasure to be here, and today I will be speaking a little bit on the HPP food safety part, particularly on the concept of the microbial validation or challenge studies. First, I just want to briefly provide a background on the current status of the HPP industry, how it has evolved. Well, it all started at a commercial scale in the early 1990s in Japan with citrus and fruit jams. The exponential growth phase of the market started late in the 1990s in the USA with the avocado products, early 2000s with meat products. And as you can see over here on the table below, in which we have the global fruit production or the estimates for year 2019, HPP has become quite a varied, quite versatile technology. So you have everything from juices and beverages, meats and avocado, which we already mentioned, seafood, fresh dips, salsas, salads, such as hummus, fruit and vegetable products, such as baby food purees, ready meals, dairy, and recently there has been a great interest for pet food, so we shall see the number of applications for this particular segment growing in upcoming years. So all in all, you can see over here on the table that global estimated production for HPP foods will be close to 4,000 million pounds or 4 billion pounds only in 2019. And well, the most outstanding feature is that in 30 years of commercial existence, there has not been any outbreak related or associated to HPP foods. So it has so far a clean record. And just to put this something into perspective, well, if we just actually quantify one case for the juices and beverages, we take down the juice and beverage global production back from the previous slide, 583 million liters produced in 2019. We break down into 16-ounce bottles, and this totals 77 million bottles of juices consumed in 2019. So one infection case, or actually less than one infection case, divided into 77 million bottles, it gives you a probability of less than 1 in 30 million to acquire an infection on an HPP juice. So you actually have better chances to win the lottery than acquiring an infection from HPP juice, which is, again, pretty outstanding. And well, this is supported by a survey carried out recently by the Canadian Food Inspection Agency. You can see that in a period of slightly over a year, the CFIA sampled over 1,200 HPP juice samples. They went to retail, to supermarkets, retrieved the samples, and then tested for different microbial quality indicators. Over here, you can see satisfactory levels would be the absence of pathogens like Salmonella, Shigella, E. coli, generic coliforms as a microbial quality indicator, and they also tested for some common foodborne viruses and parasites. You can see that satisfactory, again, would be non-detection of these pathogens. On the left, you can see the results over here for the summary for the bacterial pathogens. All HPP samples had satisfactory results, and the same applied when they analyzed for either viruses or parasites. So again, all HPP samples were free out of pathogens from samples obtained from retail stores. And just to give you an idea what type of products they analyzed, 30% of the juice samples corresponded to fruit juices only. Only 2% were pure vegetable juices. And the majority of them, as you can see, it's a mix of or a blend of fruit and vegetable juices. So again, it's pretty outstanding. 1,200,000 samples spread over a year and no pathogen detection. So this is a thing of an every day for the HPP, where processors use that technology to deliver safe food for consumers. Despite the success, well, there's still kind of absence, or there's still not a consensus on what's defined as a safe harbor, and all HPP products going into the market need a validation. So safe harbor, we have the definition over here, a recognized procedure that can be employed with further validation studies. So now to get this a little bit into context, that 30 years of industry, we certainly know what can work, what doesn't, for lots of different food products in terms of the pressure level, holding time, formulation factors such as pH, water activity, that can certainly give you a greater chance of success applying the HPP technology. But still, we need to get further details and know the exact breaking points. So again, just to put it into a different context, it's like having a minefield. We need to pinpoint exactly the locations of the mine so that we do not step over and get blown up. So as of now, the HPP industry has been over here in the blue line. But again, we need further detail regarding understand how the different parameters of the HPP cycle, formulation, storage conditions, and all those laboratory protocols used to adapt pathogens for the validation interact. And again, that this will give us an idea on where the technology fails, and that way we can create a very broad safe harbor way in which we will not require further validation. So this is a very complex task, actually. Over here under the microscope, it's a very diverse, very varied world. So just like humans, pathogens and bacteria have a different likes. So some of these will be able to adapt to cold conditions, others prefer more warm weather. Some will like salty foods, other acid foods. So there's a great variety, and in order to establish the safe harbor, we need to kind of filter these different varieties or subtypes of bacteria. This part of this was done by Hypervaric for my colleague Mario Gonzalez, which was part of his doctoral dissertation. He basically evaluated different over 30 plus subtypes for pathogens such as E. coli, Listeria, Salmonella. He evaluated, isolated, placed it into media, evaluated the pressure resistance, and not only pressure resistant, but also the capability of these different subtypes to recover during storage. So the ones with the highest risk can be over here observed in the red squares. And in addition to all these variables that I just mentioned, the capability to adapt to the media, HPP resistance, you also need to take into account whether some of these might be actually hazardous for bacteria. So it's really hard to combine all these variables, and that's part of the work that needs to be done in order to establish the safe harbor. Well, then that's not the greatest challenge. The greatest challenge is actually that under the laboratory, we can actually create or design a very, very worst case, a scenario that is very likely to fail. But also you need to balance out and wonder whether these scenarios you are carrying out under the laboratory have of practical importance, whether they can be actually found in the industry. So that's the biggest challenge, trying to balance between the fundamentals and the industrial practicability. And in this case, well, trying to cut out the excessive details, going perhaps to the worst case, to the lower case, or the other way around. And again, it's difficult, but try to keep it as simple as possible, but oversimplifying the approach can also yield a hazard for consumers. So overall, it's again a work in progress in which we are glad to take part. There's no right or wrong answer as of now. It's just that based on the science facts we obtained from different experiments, experts from the industry, academia, governmental agencies need to get into agreement and see what can be done in order to establish a safe harbor for HPP juices and other foods. With all that, again, as of now, the current status, all products going into the market need to go through a validation study, which is what we'll be now covering in this presentation. First, let's just remember that HPP is just a part of your hazard plan. hazard analysis, and critical control points, as observed here on the title. So for juice processing, you have different steps, everywhere from receiving the raw materials, storing the materials, washing, cutting, juicing, bottling, HPP, and cold storage. So there's different risk in each of these steps. You have, in example, physical hazards, which can be twigs, stones, debris that can be easily removed during storage or while culling the raw materials. You have chemical hazards such as pesticides, in which you can get a certificate from suppliers that the raw materials are comply with the required pesticide levels. And in this case, HPP is used to control or minimize the risk associated with biological hazards. We just saw from the survey from Canada, it can be bacteria, viruses, or parasites. So HPP becomes a critical control point to minimize the biological hazards, and the parameters such as pressure level, holding time, become your critical limits. So you need to validate whether the conditions you apply yield a safe food. And just for everyone, every entrepreneur out there, that hazard knowledge is mandatory. If it's not you, you should have someone in your team that's familiar with this concept in order to keep the safety for the food production. So training is strongly advised. Now jumping into the validation study. So it's just a series of experimental experiments carried out or designed by a process authority, which is an individual with a sufficient expertise in terms of HPP and food safety. And you will basically evaluate the different aspects of food processing that might influence the growth and adaptability of foodborne pathogens. From the HPP part, we have the HPP cycle parameters, pressure level, holding time. Temperature can sometimes be considered as a critical parameter as well, particularly when you're combining high pressure with moderate or high temperatures, which will be covered further in our HPP Innovation Week, the application finally of this application at a commercial scale. For formulation parameters or properties, you have the pH, wear activity, percentage of salt, sugar, fat, protein, so different components that may affect the way pathogens adapt or that they take these as resources to recover after the HPP. And finally, the temperature-time combination that keeps the food safe. So all of these are the main factors that are being evaluated in an HPP validation study. And the validation study starts when you ship fresh samples to the laboratory and then places in direct contact with the bad guys shown over here on the screen. Pathogens, bacteria such as Listeria, E. coli, Salmonella are among the most common foodborne pathogens. They allow the pathogens to grow to a non-concentration, a very high concentration that should never ever appear in a food processing facility, but acts as a worst case scenario. In this case, just to provide a number, it's like about 10 million cells per milliliter or per gram of food. So now the inoculated samples are taken into the HPP facility, processed, and you can see over here that most cycles within the industry take place in the 4,500 to 6,000 bar range, holding times between 1 through 6 minutes, and it uses chilled or room temperature, 4 to 25 Celsius. So before the HPP, we mentioned that we had 10 million cells per gram or milliliter of food, which corresponds to 7 units in a log 10 scale. And over here on the left, you have the table to observe that 10 cells per milliliter or gram will correspond to 1 unit in the log 10 scale. You multiply by 10, it's 100, correspond to 2. Multiply by 10, 1,000 corresponds by 3, and so on until we get to the 7 log units. So for raw juices and other raw foods, a 5 log reduction, a 100,000 tenfold reduction of the pathogen population, it's been used as a food safety standard. So for the pathogens, again, we will have 1, 2, 3, 4, 5 log reductions, and preferably get rid of all of those, get non-detectable levels. This is because, remember, you also need to monitor that pathogens do not recover during the shelf life to make sure that 5 log reductions or non-detectable levels remain during the duration of the shelf life, at least. So a good practice is to actually go beyond the target shelf life, just in case that you have an absent-minded consumer that has the food product back in the fridge, gets it over after the expiration date and decides to consume it. So just That's a good practice for the validation studies. So at the end, well, the process authority summarizes the results, evaluates whether they apply the HPP cycle conditions, formulation parameters, and storage are safe for consumers. And if this is the case, then the validation study is incorporated into your HACCP plan. So it states the minimum pressure level, the minimum holding time, the maximum pH in which the food can go in order to keep the conditions safe for consumers. So before actually engaging into a validation study, the best practice is to actually carry over some preliminary studies. This is to determine what would be the shelf life in terms of a quality perspective, as you can have changes in terms of the appearance or the flavors. And if it doesn't meet your expectations during or for your target shelf life, you can always go back to the kitchen and reformulate as many times as needed. Please be aware that, well, if you actually do the validation study and then carry over and need to reformulate, then adding or retrieving ingredients for the formulation actually changes the way pathogens adapt or survive the HPP. So you will maybe throwing money away. You might need to carry over the validation study. So always try to get these preliminary studies first. The other thing of the preliminary studies is that HPP is not a sterilization technique, implying that some pressure-resistant spoilage microorganisms that do not pose a threat to consumers can actually recover, such as lactic acid bacteria. And well, there can be also a good indicators on how pathogens would behave after the HPP. So it's also good from that standpoint as well. Another good practice, best practice, is to validate each and every one of your products if you have the resources. This might not be the case, particularly for small, medium enterprises. So FDA allows to group products with similar formulations under what is being called the umbrella study or a single validation study. So you basically take the worst case scenario, the fruit product in which pathogens are more likely to be present, in which pathogens are more likely to survive after the HPP. And so just by common sense, if the worst case scenario, it's actually good to go with HPP, such will be the case for the rest of the formulations. And well, important to mention that the process authority can be someone within your staff or external, as in our case, both panelists participating here should be the ones guiding you through the validation, through the umbrella approach. So just to take this into account. And just to get into the end of the presentation, we have a safety ensured, tasty and good looking food products. You are all set to hit a market, perhaps with a hyperbaric HPP units, as we share nearly 60% of the installed units throughout the world. So this has been all by my part as of now. I will be available at the end to answer any questions that might have. And well, as of now, I would like to kindly share the presentation reins to Randy. Randy, if you can please step up and go on with your presentation, and thank you for participating.
Okay. Thank you, Vinicio. And thank you, Hyperbaric, for giving me the opportunity to give a presentation on some of the activities that we've been performing at the Cornell University High Pressure Processing Validation Lab. So the Validation Center that we developed actually started in 2016 in terms of identifying the space, working with companies, and to get to the point where we were at, we actually, it was a true collaboration. We had government funding, Cornell put in money. It wasn't only the unit that was costly, but because we're working with biohazardous level 2 pathogens, we had to have special containment areas and separation from the lab when working with any of the products. And we actually, it was a collaboration between academia, government, and private industry. We received funds from 3 food companies that help support purchasing the unit, as well as providing some assistance for the renovations. And this actually shows all the parties that were involved at the ribbon cutting. This was February 2017, so it was quite monumental because the actual reconstruction of the facility and bringing in the unit was achieved in less than a year. So the Cornell HPP Validation Center actually has 2 prongs. So we the first objective that we do is we provide HPP validations for the food industry. We do pathogen challenge studies, as well as, like Vincencio said, that you need to know the shelf life of your product. So we provide shelf life determinations as well. Then we have the research side, and this is really to provide the research and data that will be used to generate the microbial safe harbors that will open it up and reduce the amount of validations that will actually be required. We also use it for companies, not only food companies, but other applications of high pressure processing. We allow them to actually bring in their product, and we will run it under different conditions, and they can evaluate it, but not leaving the laboratory, because in a biosafety lab, you're not allowed to have any product that leaves. So our biggest effort is actually performing those pathogen validation studies. And like Vincencio said, the validation report is specific to each of the products and the company. So because there's even minor variations that can result in differences in terms of the response of the pathogens to the conditions of the HPP treatment. And just like Vincencio said, any modifications in the formulation may require revalidation. So what I tell companies is make sure that your final formulation before you actually send it for pathogen validation studies. And then, like Vincencio said, there is the possibility of grouping very similar products or selecting a worst case scenario in terms of the formulation. But the regulatory agencies can closely scrutinize your grouping and your selection of your worst case scenario. So I would highly recommend having someone that's well trained in food safety and food microbiology to assist in identifying which products you're grouping together. So it's in terms of the pathogen response, it depends on the formulation. So, and what that actually is your intrinsic or physiochemical properties, such as pH, Brix, water activity, salt content, sugar content, and then how it's actually handled before HPP treatment, as well as even after. So, and it's very important in terms of identifying which pathogens you actually validate your product with. Now, what we do is we have to have commercially produced product. So that's essential, because it has to represent the typical conditions that the food would be produced when it's actually going to be HPP processed by the industry. It has to be in a flexible container. We then look at the product and we select isolates of the pertinent pathogens that are close to outbreak strains that we have in our collection. So for the most part of the validations that we perform, we usually choose Listeria monocytogenes, E. coli 157, and Salmonella. And these are all 5-strain cocktails. We then process them under the pressure and the time that the company wants to have as their critical limit for their HACCP plan. So I would say that the majority of companies process at 87,000 PSI. And so we actually do the validation study at 85,000 PSI, because that's your critical limit, is what we actually do the validation under. And the 87 is your operating limit. So if we conducted the HPP validation study at 87,000 PSI, if you dropped even 1 PSI below that, it would require corrective action. So it provides the processor a little bit more wiggle room in terms of, in case you have any kind of variation in your pressures. We then analyze it using standardized FDA bacteriological analytical methodology. Now, as Vincencio said, those samples have to be analyzed at least 2 times over the shelf life of the product. Plus, you have to go, FDA suggests going 50 to 100% additional time to make sure that pathogen regrowth does not occur. Then once we see that we've achieved a 5-log reduction, there is no regrowth, So all those conditions to achieve that 5 log reduction are given in a formal validation report that is included in your HACCP plan, as Vinicio said. Since February 2017, we've actually conducted over 500 pathogen validation studies, and you can see the breakdown in terms of the different types of food products. We have had a lot more pet foods coming in within the last year, but for the large part, it's a lot of fruits and vegetables and their products, so purees and juices and anything like that. We have also performed over 700 shelf life determinations. So companies usually have a general idea in terms of what their shelf life they would like it to be. And so in the requisition form, they put down what the shelf life is, and then we take it out for additional 50 to 100% of that shelf life to observe for when the actual shelf life is actually ended. Now onto the research side, when we started, a lot of the data that was generated on HPP and activation of various microorganisms, including pathogens, they were conducted on small units, and like 250 mil up to 1 liter chambers. Now the pressurization and depressurization is very, very different than a commercial unit. So that was one of the things why the validation center had to be built, was because it's actually a commercial unit. So using the commercial unit, we wanted to look at what we identified as critical factors for the HPP process. Of course, it's going to be your pressure and time, but also the influence of pH. Not only pH, but the type of acid that is actually in that food product, water activity, and then the water chamber temperature. Now, one of the first things we did was look at those 3 most common pathogens that we use to perform the validations with: E. coli 157, Salmonella, and Listeria monocytogenes. And we wanted to see, basically to determine the pressure resistance, you look at the slope of the inactivation lines for the different pathogens. Each of these pathogens were treated at 3 different pressures: 400 megapascals, which is the red line, 450, which is the blue, and 475, which is the green. Now, the sharper the slope, such in Salmonella, shows that it actually is less sensitive compared to E. coli 157. And it was known before that E. coli 157 H7 is actually more of those. It's the most pressure resistant. What we also wanted to look at was the water chamber temperature, because some of the regulatory agencies were wanting to have the water chamber temperature as a critical control point. For acidic products, pathogens actually die off faster due to acid exposure under warmer temperatures. So to show that chamber water temperature isn't so much of a factor, we actually treated the 3 most common juices that are consumed in the United States and probably around the world. And what you can see is, if you look at grape for Listeria, which is the last 2 bars on each of the graphs, the water chamber temperature didn't make any difference. Now, if you take a look at grape and apple juice, and you look at E. coli 157 and Salmonella, so E. coli, you can see that it is actually inactivated faster under these at 20 degrees Celsius compared to 5 degrees Celsius, and under 550 megapascals for 1 minute. So in Salmonella, it's in apple juice, it was inactivated. So you can see down here in apple juice, Salmonella was inactivated faster at 20 degrees Celsius compared to 5 degrees Celsius. So, but what this is showing you is that what we were seeing here was that there's a contribution of acid type that is influencing the inactivation rates and differences between the pathogens, with the exception of Listeria, which is sensitive regardless of the acid type. So what we did was to look at just the effect of pH. We took those 3 juices and we neutralized them so that they all had the same pHs. So you can see that grape juice had an initial pH of 3.4, apple had 3.7, and orange had 3.7. The black line at the top of each of these three is actually Listeria monocytogenes, and it was inactivated at the same rate regardless of the pH, indicating that it was very sensitive under acid conditions to HPP treatment. What you can see is that with orange juice, as soon as you increase the pH, you dramatically reduce the amount of reduction. Apple juice was less, but grape juice also showed that as soon as you increase the pH above its normal or expected pH range for the unadjusted pH, it makes a huge difference. So you go for grape juice at 3.4, we achieved a 7 log reduction, but at 4.5, we only achieved a 1 log reduction, indicating that pH and the type of acid plays a huge role. Another thing that we did early on was one of our first large validation studies was for a company that was making purees, and we saw large variations in terms of pathogen inactivation between the different types. There were different acids, but when comparing same acid type products with the same pH, we saw unexplainable differences, and we specifically looked at the water activity, and we saw that anything that was about 0.96 and below, we were getting very little pathogen inactivation. Prior to this, no one had really looked specifically at water activity at these higher levels, but it was known that when you get down to a water activity of 0.7, that definitely HPP activation was very, very poor. So what we did was we took two different types of acid at the same pH, and we tested with these different pathogens, the first one being E. coli 157. So you can see before HPP, we're starting with 7 log, and at water activity of 1, which is just water, we saw complete inactivation and we saw no regrowth. As soon as we adjusted the water activity to 0.98, we saw a dramatic reduction in the pathogen inactivation. And you can see here that there was actually less inactivation with citric acid compared to acetic acid. Acetic acid is a much stronger organic acid compared to acetic, malic, or tartaric. And then once we got to 0.96, we basically did not see any inactivation. So this was huge in terms of a discovery for the field. Same thing, acetic acid and citric acid, 3.5, citric being on the right-hand side. With Salmonella strains, same thing with water activity of 1, we saw complete inactivation. 0.98, we saw a reduction. And then 0.96, we saw basically very little inactivation. And then when you see the gradual additional decrease, that's actually due to the acid that's contained in the product. For Listeria monocytogenes, it was the same trend where with Listeria 0.96, we did see some reduction. And remember, Listeria monocytogenes is the most sensitive of the 3 pathogenic strains. But below at 0.94, we saw no inactivation. So it still is very concerning. So water activity for a product, you should be checking before you do a validation to make sure that you have success with your HPP. The other thing that we found was very, very important is the selection of the strains. We actually spent 3 months gaining a collection of different pathogens from different food products. So we have vegetable strains, we have dairy strains, we have seafood strains, and we have meat strains. And we had a company that had already validated several of their products, and they came to us to do an additional validation for essentially the same product. And they had received greater than 5 log reductions with another company. But we did not achieve a greater than 5 log reduction. We were trying to figure it out, and we found out that the company that was performing the validation studies for them on their vegetable products were using meat strains, And so we looked at the influence of the strain type in terms of HPP inactivation. So we did meat strains and then we did vegetable strains. And you can see there was a 6.15 log reduction achieved with the meat strains, but with the vegetable isolates, we only achieved a 4.13. So what we do as a standard practice is we use a 5-strain cocktail of isolates that are from the actual type of food. So if it's meat, we use meat strains. If it's a fruit or vegetable strain, we use those strains, and same for dairy, because it makes a huge difference in terms of their response to the HPP treatment. So just sort of breaking it down in terms of what are important experimental parameters that you have to take into consideration when performing a validation is you have to use a commercial processing HPP unit. The strains have to be or should be from the same type of origin as what the food product that's going to be treated. They should be prepared properly too. So if it's an acid product, the pathogenic strain should be acid adapted prior to inoculation. We also minimize the time between inoculation and the HPP treatment because, especially for acidic products, the acid will actually sensitize the pathogens, and you'll get more of an HPP inactivation result than if you rather than if you minimize that time between inoculation and processing. We also minimize the time between processing and plating. This is because we want to see the actual HPP inactivation and not due to any inactivation that might be assisting in pathogen die-off in addition to the HPP, like acid. It could also be certain byproducts that microorganisms produce can actually result in pathogen inactivation. And then, as I said earlier, we actually have to test for pathogen regrowth anywhere between for 150 to 200% of the actual shelf life. With that, if you're interested in contacting us for a validation, there's the contact information. The key person is Andy Humiston. There's the address, and you can actually go to the website and fill out the requisition for a validation study there. And with that, I will turn it back to Jorge.
Hey, thank you, Dr. Sarment and Dr. Guarabo, for your presentation. We'll have some questions coming, and we'd love to share with you. Either one of you can answer the question. The first one we have here is that how far are we from establishing a safe harbor for HPP juices and other foods?
I'll take that one. The reason why we actually did apple, grape, and orange is because it's the majority of the juice that's consumed in North America and probably around the world. We've done extensive work looking at the influence of pH, the water activity under normal ranges of apple, grape, and orange, and the results are very consistent. So I would suspect that the safe harbors for those for fruit juices are established. Vegetable, on the other hand, there needs to be additional work because vegetable juices typically are higher pH. But there are some fruit that have higher pH, like mango and stuff. So I would say that additional work may be needed on some of the fruit juices, but and definitely more for the vegetable. For other food products, it's going to take additional research, especially with the diversity of foods that are being high pressure processed now. Everything under the sun seems to be high pressure processed now.
So yeah, I would just like to add that, again, it's no coincidence, 30 years of commercial applications for the HPP industry, no outbreaks, is relatively safe. But as we both shared research advances from Cornell hyperbaric, we still need to tune in and try to figure out more details in order to establish the exact failing points which is needed for the the application of the safe harbor, and we shall see more of this coming in recent years.
Great. Talking about next steps, what will be the next research step for the HPP validation, either for juices of other food?
So for juices, I think it's we have to look at more the tropical juices like acai and guava, mango. And basically doing what we did with the orange apple and grape is looking at what the normal range is depending on the variety of the fruit, looking at what's the highest pH that occurs, and looking at the pathogen response under different pressures like we have. For other foods, I think you almost have to prioritize in terms of the volume that is actually being high pressure processed and group those and then do actual pathogen studies under varying conditions to see what the response of the pathogens are.
Good. And we all know that food regulation has a lot of variables. Do you foresee any changes in HPP, juice or food regulations in the short term?
So I think with the exposure of the number of toll processors or the number of HPP units that are in the food industry, the regulatory agencies are actually becoming more familiar and more comfortable. And definitely FDA had, well, no, I would say that all the regulatory agencies in the United States have opened up for alternative processing methods compared to the traditional thermal.
Yeah, for sure. As the technology becomes more familiar, more widespread between both consumers and processors, we shall see this growing. And also with our research advances, hopefully we will see soon some more generic guidelines in terms for the industry and HPP foods in particular.
Great. Hey, I don't mean to change gears here, but we do have a lot of interest, a lot of questions regarding the commercialization and validation of HPP milk. What are the main challenges holding back HPP milk in the USA?
So yes, I've had a lot of inquiries from various dairies for HPP treated fluid milk. The problem is that the pertinent microorganism or pathogen for milk is actually Coxiella burnetii, and the pasteurization temperatures are actually based on Coxiella burnetii. Coxiella, you cannot grow on synthetic media like you can for Listeria, Salmonella, Bacillus, and Vibrio and Shigella. You actually need to have animal models, and it's actually a next level up in terms of biohazard. So every time you go up in terms of a biohazard to work with it, the price goes up, and especially animal models. It takes a lot of animals to actually do those studies. So if someone's got enough money, it can definitely be performed, but that's going to be the biggest hurdle. So it's not your typical Listeria, Salmonella, and E. coli that you use.
Well, it's Coxiella, and perhaps you also need to screen for the other pathogens as well. And I think with milk is also a very challenging system. So it has all the resources that either protect or allow bacteria to recover proteins, fats, minerals. And in fact, there are a couple of companies doing it in Mexico and in Australia and New Zealand that rely more on the absence of pathogens prior to going in the HPP, though. So they use it as a shelf life extension step. And there's the next big stage will be the combination of high pressure with mild temperature or high temperature. We will actually allow to yield or achieve the food safety objectives, not only for the pathogens, but also for some spore-forming pathogens such as Bacillus cereus, which is of concern in milk products as well. And that will be also part of the future applications for milk, I guess.
Yeah, it's very concerning in terms of the number of foodborne illness outbreaks that have occurred with raw milk. And the reason why people want to drink raw milk is because they think that the thermal treatment changes the properties and the probiotic properties of the milk. So with HPP, it would definitely enhance the safety for what I consider to be a high-risk product.
Well, thank you very much. And there's certainly a lot more questions, but we try to keep these sessions in a regular time. But you can definitely reach out to both of these gentlemen. You could tell that there's a lot of knowledge coming from these two personalities here with a lot of studies and a lot of strong background. And most importantly, let me tell you, the audience, that they are at your disposal. You got their information there. If not, you can reach out to us at hyperbaric.com and we'll get back to you. That's what we do. That's our passion. Let us have the opportunity to work with you in your next HPP validation project. HPP treatment could be a game changer for your product and your company. Please reach out. Let us be part of your next successful move. Thanks again for trusting Hyperbaric, and thank you for your engagement in the HPP Innovation Week. Stay tuned. A lot more to come. We're looking forward to see you all for next sessions. Thank you very much.
Thank you.
Bye-bye.